Hello, everyone. My name is Jackson Brindle, and this is our group presentation on bladder cancer. Our group consisted of Martha, Toneasia, Madison, and myself. Basic cancer research. 4 types of bladder cancer based on cell or tissue origin is recognized. Bladder cancer often occurs most frequently in transitional cells that make up the tissue layer lining the bladder and stretch when the bladder is filled with urine. This is called transitional cell carcinoma or urothelial carcinoma and comprises of 95% of bladder cancer diagnosis. Roberson is one of the many drugs used to treat this type of bladder cancer. Another type, which is much less common and occurs in 1 to 2% of diagnosis, is squamous cell carcinoma. This is associated with chronic irritations such as long-term catheter use, UTIs, or in countries of lower socioeconomic status due to likely contact with parasites and bacteria in unsanitary conditions. Other less common types include small cell carcinoma and adenocarcinoma. Adenocarcinoma occurs in the mucous cells of the bladder. Bladder cancer also encompasses multiple cell types and exhibits tumor heterogeneity. Tumors are measured in terms of differentiation or grading. This looks at how different the tumor tissue is compared to normal tissues. Normal tissue is comprised of various cells working together or well differentiated. Grade 3 tumors are poorly differentiated and are the most serious type. Tumor types are also determined by the relation to urothelial and invasiveness in the muscle cells, as we will discuss later in staging.
RAS association domain family 1, better known as RAS F1A, is a tumor suppressor gene found on the 3rd chromosome. As a part of the RAS pathway, it performs signal transduction in a cell. The figure on the right visualizes RAS F1A signal transduction pathway, which aids in cell cycle control, maintenance of genomic stability, cell motility control, microtubule stabilization, and apoptosis functions. The hallmark of RAS F1A is genome instability. This is due to hypermethylation of CpG islands in the promoter sequence, which prevents signal transduction to occur within the cell. Without communication from RAS F1A, the cell loses the ability to control the cell cycle, maintain genetic stability, enables cell motility, and has resistance to apoptotic factors. There's no heritability component because the addition of methyl groups by cancer cells happens randomly. When the methyl group is attached to a promoter sequence, it silences gene transcription, which is passed on to daughter cells only, but not the germline cells. Fibroblast growth factor receptors, also called FGRs, is the proto-oncogene that is targeted by erdafitinib. FGRs is a group of transmembrane receptor tyrosine kinases with 4 receptor isoforms. These proteins are found on the cell membrane. The figure on the right shows the 3 domains that comprise FGRs. The extracellular domain has 3 immunoglobulin domains that compose the binding site for the fibroblast growth factor, which is a ligand for the receptor. The single pass transmembrane domain enables transduction to the intracellular tyrosine kinase domain. The intracellular tyrosine kinase domain phosphorylates tyrosine residues that leads to the cascade activation of receptor proteins. This ultimately leads to cell proliferation, differentiation, growth, and migration. The hallmark for FGR is sustaining cell proliferation through autocrine and paracrine signaling. The missense mutations in the FGFR3 affect 84% of patients with urothelial cancer. The missense mutation in S249C changes the amino acid TCC to TGC, causing disulfide bridges to rearrange due to the new cytosine residue. The addition of this disulfide bridge allows the extracellular domain of the receptor to not be as selective and allow the cell to polymerize without the FGF ligand being present. This leads to amplification of more receptors through cell proliferation. The FGR TACC fusion comes from a fuse of the FGFR3 immunoglobulin, single pass transmembrane, and tyrosine kinase domains to the TACC3 coiled coil domain. This fusion keeps the kinase protein active, which leads to cell proliferation as well.
Okay, so now we're going to talk about bladder cancer epidemiology. So these are some of the public health implications of bladder cancer. So the National Cancer Institute estimates that the number of new cases of bladder cancer in 2021 will be approximately 83,730, leading to an incidence rate of approximately 0.025% and making it the sixth most common cancer type overall. Of those 83,730 diagnoses, men will make up the majority of those new cases at approximately 64,280, while women are only estimated to account for 19,450 in new bladder cancer diagnoses in 2021. In North Carolina alone, it's estimated that approximately 2,650 people will be diagnosed with bladder cancer by the end of 2021. Bladder cancer is estimated to account for approximately 4.4% of cancer diagnoses within the year. The National Cancer Institute also estimates that the projected number of bladder cancer mortalities in 2021 in the US will be approximately 17,200, making up about 2.8% of cancer deaths total. It is projected that approximately 12,260 of those bladder cancer deaths will be in men, and the other 4,940 will be in women. After the 1970s, bladder cancer incidence seemed to be on the rise, but finally in 2008, there seemed to be somewhat of a slow decline in bladder cancer incidence, approximately dropping 1% each year until 2017. Incidence rates for people of white ethnicity in 2018 sat at approximately 22.9 per 100,000, whereas incidence rates for every other ethnicity in 2018 sat in between 8.8 and 11 per 100,000, which is a dramatic reduction. So these are just some more public health implications. The overall 5-year survival rate from 2011 to 2017 was 77.1%. However, there was some variation in survival rates when it came to where the cancer was located. If it was only in situ, the rate was about 96%. If it was only localized, it was about 69.6%. If it was regional, it was 37.5%. If it was distant, it was 6.4%. And if it was unstaged, it was about 48%. I created this graphic with that information just to help you see how that breaks down visually. There's also some disparity in 5-year survival rates with different races, with 5-year survival rates for individuals with Native American, Asian or Pacific Islander, Black and Hispanic ethnicity sitting lower than the average at 63.7%, 74.9%, 64.6%, and 72.7% respectively, whereas people of white ethnicities sat above the average at 78%. As of 2018, approximately 82.5% of people living with bladder cancer were above the age of 65. So these are the risk factors for bladder cancer. The most prominent of these is, just like many cancers, is smoking and tobacco use. And then other risk factors include age, gender, and workplace conditions. Bladder cancer is actually one of the most common consequences of smoking and tobacco use, and smoking is responsible for approximately 50% of bladder cancer tumors. This high number may have something to do with the fact that smoke contains aromatic amines and polycyclic aromatic hydrocarbons that have been proven to have a strong link to urinary bladder cancer. Going back to the previous slide talking about prevalence rates, men are far more likely to get bladder cancer than women, which might be linked to the high rates of men using tobacco compared to women. The decrease we have seen in the past few years in the incidence of bladder cancer could be linked to the shift in attitude about smoking and tobacco use from accepted and encouraged to something seen as unhealthy and discouraged. As smoking becomes less and less accepted, it is reasonable to assume the incidence rates of bladder cancer will too. Public health campaigns focusing on the relationship between smoking and bladder cancer could be good ways to lower the incidence rates and mortality rates of bladder cancer. Not only could these campaigns potentially lower bladder cancer incidence rates, there could also be many other potential positive side effects for health benefits as well. So this is just a graphic showing the different risk factors of bladder cancer, the major ones at least. And like I said, gender, age, smoking, and then workplace conditions are the biggest ones, and smoking is the biggest.
Hi, I'm Toneasia. I'm focusing on the intervention study part of the our presentation. So the name of the study was erdafitinib in locally advanced or metastatic urothelial carcinoma. Another name for the drug is Balversa, so I'll probably be using that name instead. It's a little easier to say. So this was a phase 2 study with a total of 99 patients. In the group, they went through an average of 5 cycles of Balversa in about 5.3 months. The starting dose was set at 8 milligrams daily, with an adamant that they could go up to 9 if that treatment was necessary. And the average follow-up duration for the survival was 11 months, with the 95% confidence interval of about 9.1 to 12.2 among the patients in the group. Of the 99 patients, 41 had the drug up to the 9 milligrams. 13 patients continued treatments for at least 4 weeks after the disease had identified as progressing. Of the 97 patients who underwent at least one disease evaluation, about 77% had a reduction in the target tumor diameter, and about 49% actually had a tumor reduction of 30 to 100%. The primary endpoint for this was the confirmed response rate among patients, and the confirmed response rate to the erdafitinib therapy was 40%, 3% with a complete response, and a 37% with a partial response. Among the 22 patients who had undergone immunotherapy before they started this regimen, the confirmed response rate of them was 59%, and the use of erdafitinib was associated with a objective tumor response in about 40% of the patients who had a locally advanced or metastatic urothelial carcinoma. As I said, the other name for this is the Balversa. This is a fibroblast growth factor receptor inhibitor, or selective tyrosine kinase inhibitor. It specifically targets the FGFR from 1 to 4. And it inhibits the renotubular FGF23. This has a kind of narrow therapeutic index. In the first study, they had up the maximum dose to 12, which was the dose limiting toxicities persisted. So move this to 9 milligrams in the second phase, which is this. And then all 99 patients reported having an adverse event in the course of this treatment. This is where this chart comes in on the side. 13 patients actually discontinued treatment because of adverse effects, including detachment of the retinal pigment epithelium, hand-foot syndrome, dry mouth, a skin and nail effect. It was kind of like unspecified. And a dose reduction was actually required in 55 patients. The most common adverse effect leading to a dose reduction was stomatitis in 16 patients and hyperphosphatemia in 9 patients. There was a reported death, but it was said to be unrelated to the drug and to the whole treatment.
Clinical bladder research, clinical course and manifestation. The staging of bladder cancer uses 3 variables. Measurements are required during exploratory surgery or cystectomy. These variables are attached to stages that represent the course and typical proximity for spread of bladder cancer when untreated. Location of the spread influences the staging as well. SEER measures 5-year survival based on extent of spread in terms of location spread by local, regional, or distant. To better understand staging, the top of the chart, staging 0 in blue, is someone without cancer. A variable with a 0 attached indicates that there is no tumor, spread, or metastasis of the cancer. Stage 0A and 0IS are the initial cancer cells in the beginning of bladder cancer. People in these stages have the greatest chance of survival. The first stages are governed by tumor growth within the bladder wall or localized. Spread to nearby lymph nodes follows complete invasion of the bladder and marks the escape from the organ or regional spread. Lastly, the cancer cells that originate in the bladder spread to distant organs and tissues or metastasis occurs. Localized means cancer cells are still contained within the bladder structure from the lumen to the adipose tissue. Here you will see a diagram of the tissues affected by various stages of bladder cancer. The further this cancer travels through the bladder into regional areas and eventually into the spread into distant organs and tissues, marks the clinical staging increasing as well. Treatment protocol and combinations. The treatment protocol for bladder cancer is influenced by the stage and spread of cancer. Transurethral resection with fulguration is the primary treatment for early stages of bladder cancer and for diagnosis in both early and later stages. Intravesical therapy, meaning putting the treatment inside the bladder, is used for early stages. This therapy can be chemotherapy, which results in fewer systemic side effects because it is local, or BCG. Intravesical chemotherapy is usually several different types of drugs. Mitomycin is one type of drug. Also, valrubicin is another type of drug, if I'm pronouncing that right. BCG, or Bacillus calmette-guerin, is a type of immunotherapy that uses a pathogen related to tuberculosis placed inside the bladder to stimulate the immune system. As bladder cancer progresses into later stages, the treatment becomes more drastic. Cystectomy or removal of part or the entire bladder may be warranted depending on the location, stage, and size. Systemic chemotherapy goes into the bloodstream via pill form, IV, or IM injections, so it reaches every part of the body, unlike intravesical therapy that is confined to the bladder. Adverse effects of systemic chemotherapy is obviously more widespread. The drug used for systemic chemotherapy varies depending on the use of radiation with the drug. Radiation therapy consists of aiming a beam of radiation towards the cancer cells. Radical cystectomy, in addition to full bladder removal, also encompasses nearby lymph nodes removal to prevent or stop spread. This is performed in later stages in combination with other mentioned treatments. The immune system does not want to attack its own cells, and cancer cells use this to evade destruction. Immunotherapy works to make the immune system recognize cancer cells needing to be destroyed by inhibiting immune checkpoints protein. Valbursa is used for locally advanced spread stage 3A or more advanced metastasis. This drug is only used when bladder cancer got worse in or following treatment with platinum type chemotherapy. Lastly, in the most advanced forms of bladder cancer, clinical trials may be the only viable option when all else fails. Average patient experience in general on Valbursa and throughout the course of their treatment is very different compared to other cancers. Bladder cancer is often neglected compared to more popular cancers such as breast cancer. This translates to a poor patient experience. Care planning, patient involvement, care, and post-treatment are all of lower quality in comparison to other cancer diagnoses. In terms of treatment itself, it is heavily reliant on stage as bladder cancer advances, so does the strength of treatment and lasting effects following treatment. Poor patient experiences such as those ignoring the problem until it's too late, result as due to this decreased visibility. Gender also factors into this as well. Male patients tend to internalize their illness, while female patients are commonly dismissed in medicine. Valbursa has shown promise in advanced stages of cancer in individuals when all else treatment has failed. Tumor shrinkage, along with reasonable side effects in comparison, such as mucous membrane irritation, inflammation, nail changes, eye disorders, and hand-foot syndrome, have been reported with Valbursa. Post-treatment, patients' experience varies depending on stage and is typically poor in comparison with other cancer diagnoses. Patients may have incontinence and living with a urostoma, along with costs of aftercare. Patients may also have to find new ways to be sexually active or overcome living sexually inactive.
Thank you so much for watching our presentation, and I really hope you enjoyed. And these are our references. Thank you.